To all of you. Today I'm going to take DNA replication. Like already you people know about DNA, RNA, everything. Now the first thing is how DNA replication takes place. Like how the DNA, which is the chromosomes or the chromatids which are present in the nucleus, how they double themselves and how it is divided equally in two daughter cells. So like all the details of this DNA replication, I'll be telling you and how it is controlled also. So basically, you all know that it is the basis of inheritance DNA and the fundamental process which occurs in all the cells, like for copying DNA to transfer the genetic information to daughter cells. So initially it is there in the mother cell and later on it gets divided into daughter cells. So each cell must replicate its DNA before division. So now you people have any idea like which phase, like cell cycle, there are different phases, no? Like G1, S, G2 and M. These are the four major phases of cell cycle. So which phase you have DNA replication? Anybody can tell me? Which phase of cell cycle you will have DNA replication?
S phase.
Yeah, correct. It is during synthesis phase or S phase. Okay, after that it just stops, because the cell goes for mitosis after like G1S, G2, and then mitosis. So that like whatever replication has taken place, that will be divided into daughter cells during mitotic division. Okay. So this is about like, anyway, you all know that DNA double helical structure and the model which is used till now, it is Watson and Crick's model, where they got Nobel Prize as well. So this is like regarding the double helical structure, where the like bases they pair with the help of hydrogen bonds, and like there is a directionality of DNA. Like the nucleotides in DNA backbone are bound together by phosphodiester linkages, like between 3 and 5. Okay, always the 5, this is the phosphodiester linkage, which is between 5 prime and 3 prime carbon. And DNA molecule has a direction. Complementary strands run in opposite direction. So it is always like 5 prime to 3 prime and the opposite strand is here, 5 prime to 3 prime and there is a direction. Like always the opposite ends of DNA, they run in opposite direction. Complementary strands run in opposite direction. It's like this. So like the DNA replication, it is semi-conservative replication. Why it is called semi-conservative replication? Because the parental strands, they are not degraded here. In parental, like the one strand of DNA is parental strand and a new strand is synthesized. So because of that, like each new strand has a parental strand and the new strand. That's why it is called semi-conservative replication. And the base pairing allows each strand to serve as a template for a new strand. So this strand is a template strand for new strand synthesis. Here, this strand is the template strand for new strand synthesis. So new duplex is half parent template and half new DNA. So because of that, it is called semi-conservative replication. So like these are some of the features of DNA replication. It is semi-discontinuous. There is a leading and a lagging strand. Leading strand, there is continuous synthesis. This is leading strand, where there is continuous synthesis, and other strand is lagging strand, where there is discontinuous synthesis. Here, like you will find Okazaki fragments, which are later on, like because there are so many fragments, so synthesis takes place in fragments in the lagging strand. For leading strand, there is direct continuous synthesis. Okay, and then these fragments are called Okazaki fragments, which are later on joined by ligases, DNA ligases, which is the enzyme which is responsible for ligation. So there are many enzymes which are involved in the process, which I'll be telling you slowly one by one. So basically DNA replication, it requires energy. So energy of replication, it comes from, because the nucleotides, they arrive as nucleoside triphosphate. So this triphosphate is there, which is like anyway, phosphate is always for energy. Okay. So there is a DNA base, there is sugar with 3 phosphates, which provide energy for bonding. And these DNA bases, they arrive because they are coming in the form of nucleoside triphosphate. So DNA bases arrive with their own energy source for bonding. So, and the enzyme which is responsible for binding and all, that is DNA polymerase 3. So there are different kind of DNA polymerases, which I'll be telling you after some time. So for DNA replication, there is a primer required. Okay, so DNA polymerase can only add nucleotides to 3' end of a growing DNA strand. So there is a need of a starter nucleotide to make a bond, because the polymerase will be working on the 3' end of a growing DNA strand. So it requires a nucleotide to start the new strand synthesis. So the strand always grows in 5' to 3' direction. Okay, because the nucleotide, the nucleophilic attack is always at the 3' end. Because of that only, the strand always, it is a universal rule that the strand grows in 5' to 3'. It never happens that it grows from 3' to 5' direction. Okay? The template is read in 3' to 5' direction, while polymerization takes place in 5' to 3' direction. So this RNA primer, which is required in the beginning for DNA synthesis, it is synthesized by primase. It serves as a starter sequence for DNA polymerase 3. Only one RNA primer is required for leading strand. Because leading strand is a continuous strand, it requires only one primer. And the RNA primers for lagging strand depend on the number of Okazaki fragments. So how many Okazaki fragments are there? So many primers are required for the lagging strand. Okay, there are, because as I told you, anyway, I'll show you. As I told you that there are so many Okazaki fragments in the lagging strand. So RNA primer has a free 3' OH group to which the first nucleotide is bound. And these are different steps of DNA replication. First, identification of origin of replication. So like in eukaryotic chromosomes, you have number of origin of replication, then unwinding denaturation of double-stranded DNA to provide a single-stranded DNA template, then formation of replication fork, initiation of DNA synthesis and elongation, primer removal and ligation of the newly synthesized DNA segments, then reconstitution of chromatin structure. So because during S phase, it is in the form of a chromatin. So first it has to go for identification of origin of replication. It has to be identified. And after that, there will be unwinding or denaturation of double-stranded DNA. Anyway, I'll tell you with the help of some diagram after some time. Okay, so these are different enzymes which are involved in the process of DNA replication. DNA polymerases, they are responsible for deoxynucleotide polymerization. So they are responsible, polymerases are responsible for adding new nucleotide to the chain elongation and all, and synthesis. Then helicases are there. Helicase is responsible for processive unwinding of DNA. So because the DNA has to be, always it is in the form of a supercoil. So it has to be unwound first. So because of that, these helicases are responsible for unwinding of DNA. There are enzymes called topoisomerases. There are topoisomerase 1 and 2, which are responsible for, they relieve the torsional strain that results from helicase-induced unwinding. So they don't allow it to break. So these are responsible for torsional strain. Like they relieve the DNA from torsional strain that I'll show you. DNA primase initiates synthesis of RNA primers, which are required for leading strand 1 primer, and for Okazaki fragments, each one requires 1 primer. Then single strand DNA binding proteins. These single-stranded DNA binding proteins, they prevent premature re-annealing of the double-stranded DNA. Actually, because at the time of replication, the double-stranded DNA gets converted into like single-stranded ones. So there is every possibility that it can re-anneal again. So these single-stranded DNA binding proteins, they prevent premature re-annealing of double-stranded DNA. Okay. And DNA ligases are there which seal the single strand nick between the nascent chain and Okazaki fragment on lagging strain. Yeah, this is like, these are different enzymes which are there. See, you have primase, which is responsible for synthesis of primer, RNA primers, which start the like synthesis of new strands. Then helicase is responsible for unwinding of the double-stranded DNA. And this primase is responsible for these primers. Okay, these are all Okazaki fragments, like which might be, this one is lagging strand, this one is leading strand. So there is only one RNA primer in this case, and this case, there are a number of RNA primers. Topoisomerases are responsible for relieving the torsional strain. This is the parental DNA. Yeah, so like just when this DNA helicase is here, which is responsible for unwinding, then you have both the strands are there. This strand is running 5' to 3'. This one is 5' to 3' like this. And like here the primer is, this is the RNA primer here. So this strand is moving from, always like the strand moves from 5' to 3'. Always it is added in this direction. Okay, 5' to 3' direction. And here also it is a 5' to 3' direction. So DNA polymerase always, like there is a nucleophilic attack at the 3 position. That's why the chain always grows in 3' direction. Then like origin of replication. At the origin of replication, ORI we call it, there is an association of sequence specific double-stranded DNA binding proteins with a series of direct repeat DNA sequences. So like wherever you have this origin of replications, there is like the double-stranded DNA binding proteins along with a series of direct DNA repeat sequences are there. So in E. coli, the origin of replication, this ORIC, is bound by the protein DNA A. It is in case of E. coli, like in prokaryotes, not in eukaryotes. There are like similar kind of proteins, not DNA A. There are other proteins which are responsible for identification and binding to origin of replication. A complex is formed consisting of 150 to 250 base pair of DNA and multimers of DNA binding proteins. So like there is a formation of a complex which consists of 150 to 250 base pair of DNA and multimers of DNA binding proteins. So many DNA binding proteins as well as so many base pairs. This leads to the local denaturation and unwinding of an adjacent A plus T rich region of DNA. So because of the binding of these DNA binding proteins, like there is denaturation in a particular place which is rich in AT. Okay, AT rich region only starts unwinding because there are only double hydrogen bonds here. So this is in case of prokaryotes. In eukaryotes, it is a little different from prokaryotes. So in case of eukaryotes, it has been studied in higher ones also. Like this is, I'm just telling you about the yeast. So functionally similar, autonomously replicating. Basically, the basic things are similar, but there are more number of proteins which are involved in the process. So functionally similar, autonomously replicating sequence ARS or replicators have been identified in yeast cell because yeast is a eukaryotic cell. The ARS contains somewhat degenerate 11 base pair sequence called origin replication element. Okay, this origin replication element binds a set of proteins analogous to DNA A protein of E. coli. In case of E. coli, you have the protein which binds to the DNA bases, that is DNA A. But in eukaryotes, it is not DNA A, it is similar, analogous to this DNA A, which is collectively called the origin recognition complex. Okay, when it binds through this, it forms a complex which is called origin recognition complex. The ORE is located adjacent to an approximately 80 base pair. Here also, like there it is comparatively nearer because the size of the chromatin, it is quite big in case of eukaryotes, and there are many origin of replications. So here approximately 80 base pair A plus T rich sequence that is easy to unwind. This is called DNA unwinding element. Okay. So the interaction of proteins, like in eukaryotes, there are different proteins in prokaryotes, but they are analogous. The interaction of proteins with ORI defines the start site of replication. and provides a short region of single-stranded DNA essential for initiation of synthesis of the nascent DNA strand. Then DNA helicases allow for processive unwinding of DNA. Single-stranded DNA binding proteins stabilize this complex. In cooperation with single-stranded DNA binding proteins, this leads to DNA unwinding and active replication. See, it's like this here. This is in case of eukaryotes. In case of prokaryotes like E. coli, it is only that DNA A. But here there are so many proteins which are involved in the process. In G1 phase, there is origin licensing. Like G1 phase will not allow replication of DNA. So these origins remain dormant here. Then these origins are active origins. Okay, there are very few active origins when it is in G1 phase. When the cell comes to S phase, synthesis phase, that time only the pre-replication complex forms here. So there are many proteins which are involved in the process. So they all bind here. There is CDC. CDC is like cell division control protein. Then you have MCM. It is mini chromosome maintenance proteins. And you have CDTs are there, which are chromatin licensing and DNA replication factors. So, and there are like, you have DDKs are there, which are DBTF4 dependent CDC kinases. So there are different proteins which are involved in the process. They all bind to the origin replication complex, and then it forms a replisome. And then there is unwinding of DNA with the action of helicases. Okay, it is like that. The replisome is formed like this with the help of all these enzymes. And then like they identify a region which is some A plus T rich region from where the unwinding takes place. So in case of eukaryotes, it is like this is just a comparison between prokaryotes and eukaryotes. See, there is one origin of replication. The DNA is circular in case of prokaryotes. So there is only one replication bubble here. Replication fork is formed like this. Origin of replication is here. In case of eukaryotes, there are different replication bubbles, like the replication fork, it forms, like there are so many origin of replication. So the DNA replication takes place simultaneously in different places. Okay. Yeah, it is like, this is for E. coli, and where there is only one origin of replication. So it will be dividing that way. Then in eukaryotes, because there are multiple origin of replication, so replication bubbles are formed in different places. So there is formation of replication fork. The polymerase 3 holoenzyme binds to the template DNA as a part of a multi-protein complex. DNA polymerase only synthesizes DNA, as I told you earlier. Just a minute. Hello, Sandhya. Yeah, tell me something important. Paper, yeah, I'm in class right now. I'll finish my class, then I'll discuss with you. Okay? Huh? Okay, no problem. It's okay. So for formation of replication fork, then DNA polymerase only synthesizes DNA, as I told you earlier, that 5' to 3' direction always. Because the DNA strands are antiparallel, the polymerase functions asymmetrically. Yeah, it functions asymmetrically because it goes in different direction. Like one chain, leading strand and lagging strands, their directions are different. So because of that, DNA polymerase also functions asymmetrically, like it functions there as it functions here. On the leading strand, forward strand, the DNA is synthesized continuously. On the lagging strand, like retrograde strand, the DNA is synthesized in short 1 to 5 kilobase fragments, the so-called Okazaki fragments. Yeah, it's like that. There is a little blurred figure, but I think you are able to see. It's okay? Can you see it properly, the picture?
Yes, ma'am.
Yes, ma'am.
Okay. Yeah, fine. It is a little blurred, but I can't help it. Anyways, so this is like how the overall direction of replication. So the first step is helicases unwind the parental double helix. Okay, this is the parental double helix. So there are two strands. This is the parental DNA. So because of the presence of helicages, like there is unwinding of the parental helix. The second step is single-strand DNA binding proteins stabilize the unwound parental DNA. So these single-stranded DNA binding proteins, they just make a complex or they bind to single-stranded DNA so that there won't be any re-annealing. Otherwise, what will happen? Like they can go for again re-annealing, and then a replication will not take place. The leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase. So this is the leading strand. So it is going in this direction, okay, 5' to 3' end. So this is your replication fork where helicases are responsible for like unwinding of DNA, and then synthesis takes place from, this is the leading strand and this one is lagging strand. So your polymerases are working asymmetrically because the chain is moving in this direction for leading strand and this direction for lagging strand. Okay. Like the primases are responsible for RNA primer, which is required for initiation of DNA replication. Okay, so here you see there are so many RNA primers, like each Okazaki fragment has one RNA primer. Okay, here only one RNA primer is required. Then the lagging strand is synthesized discontinuously. Primase synthesizes a short RNA primer, which is extended by DNA polymerase to form Okazaki segments. Okay, like the synthesis starts from here, 5' to 3' and it goes. After the RNA primer is replaced by DNA by another DNA polymerase, DNA ligation joins the Okazaki fragments to the growing strand. So like these, because initially it is RNA primer which is going for synthesis. So it is only like the base pairing will be in such a way because there is one uracil different in case of RNA. So it has to be synthesized in such a way that later on, like DNA polymerases, they are responsible for, like, because the sugar and all is also different. So the RNA primers is replaced by DNA later on. And DNA ligase is responsible for joining these Okazaki fragments. So this is like a diagrammatic representation how DNA replication takes place. See, whenever you have such questions, always like go for diagrams, okay? Like it always gives you better marks whenever you are supporting your written material with the help of diagrams. It always gives a good idea about thing, and it is easy for you to understand the thing also when you are drawing the diagram. Explanation is also easy. So like formation of replication bubble. Replication occurs in both directions along the length of DNA, and both strands are replicated simultaneously. This process generates replication bubbles, okay, like this. So origin of replication, there are in eukaryotes, there are many origin of replications. So both sides, like DNA synthesis takes place. So there are bubbles, replication bubbles are formed. So many are there, parental strand is there, daughter strand. So these are replication bubbles. So replication fork is here, two daughter DNA molecules. In eukaryotes, DNA replication begins at many sites along the giant DNA molecule of each chromosome. So like there are many replication bubbles. In your prokaryote, there is only one. So this is replisome assembly. Like there is in G1 phase, like all these are the prerequisites for going for DNA replication. So G1 phase, all the preparations will be going on. That is called gap phase. So this is your ORC, origin replication complex. And this is like the early firing origin. There are MCM, DDK is there, CDCs. Like these are the proteins which are responsible for making this complex, okay, so that it is ready for replication. It helps for replication. So there is helicase loading, and helicase gets it starts going for unwinding of DNA. So during S phase only, all these things get accumulated, all the proteins, like the complex forms. Then there is helicase activation. There is polymerase. There are different kind of polymerases, which I am going to tell you. You have a polymerase. In case of Prokaryotes, there are only three, like three are there in case of eukaryotes also, but there are some subdivisions and all. But in case of prokaryotes, there is only polymerase 1, 2, and 3. Here we pronounce it differently. There is polymerase eta, there is polymerase alpha, and there is polymerase delta. So these are these polymerases, they also get activated, and this kind of complex forms. This is the complete replisome here. Okay. So there are a number of different DNA polymerases, molecules engaged in DNA replication. These share three important properties. So DNA polymerases has three important properties. One is chain elongation. They are responsible for chain elongation DNA. Then processivity is there, and then proofreading. So they have three main functions. One is chain elongation, processivity, and proofreading. Chain elongation accounts for the rate of nucleotides per second. Like how many nucleotides are added per second? This is called chain elongation, at which polymerization occurs. Processivity is an expression of the number of nucleotides added to the nascent chain before the polymerase disengaged from the template. So like it is in between the process, like how many nucleotides are added to the nascent chain. And proofreading function identifies copying of errors and corrects them. So whenever there is an error, because there is like the process is going on continuously, there is a possibility there is a wrong mismatch, like a wrong base pair, which is getting added. So this proofreading function is carried out by DNA polymerase only, and it corrects the errors. In E. coli, polymerase 3 functions at the replication fork. Of all polymerases, it catalyzes the highest rate of chain elongation and is the most processive. DNA polymerase 3 is the main DNA replicating enzyme, like polymerase, which catalyzes highest rate of chain elongation. Then polymerase 2 is mostly involved in proofreading and DNA repair. So this is responsible for chain elongation, polymerase 3. Polymerase 2 is involved in proofreading function and DNA repair. And polymerase 1 completes chain synthesis between Okazaki fragments on the lagging strand. So the main polymerase is polymerase 3 here, which is responsible for chain elongation. And this polymerase 2 is for proofreading and DNA repair. And polymerase 1, it completes chain synthesis between Okazaki fragments on the lagging strand. So this is for E. coli, fine. So like what the difference between, these are different differences between the polymerase 1, 2, and 3. So basically the polymerization rate, like polymerase 3, as I told you, that it is responsible for chain elongation. So the polymerization rate is high in case of polymerase 3. Processivity is also high. Then proofreading function is more for, because polymerase 1, 2 is responsible for this proofreading function. And this 3, it has the activity of 3' to 5' and 5' to 3' exonuclease activity. This is 3' to 5' exonuclease activity. This, all of them, they have, it has got both proofreading activity. The primer removal is best in case of DNA polymerase 1. And it is like in polymerase 2, primer removal is not there. So this is because this is responsible for the filling the gaps between the this lagging strand, and it is then strand synthesis also. This is DNA polymerase 1. It is like in lagging strand only strand synthesis is there. DNA polymerase 2 has no role in that. It is for both the strands. It helps for chain elongation. DNA repair, this is active, this is active. It has no role in that. So there are, as I told you, that there are different, basically there are three groups only for DNA polymerases. So eukaryotic cells have counterparts for each of these enzymes, plus there are some additional ones also. So like in E. coli, we call it DNA 1. And here mammalian cells, it is called DNA polymerase alpha. So this also have similar functions, like it also goes for Okay, thank you. Yeah, so we were discussing about the function of different polymerases and the counterparts in eukaryotes. Yeah. So DNA polymerase 2 in E. coli and similar uh, polymerases are there in eukaryotes, like in mammalian system. We have epsilon, which has DNA proofreading and repair function, because in E. coli, DNA polymerase 2 is responsible for proofreading function and all. So here also this one epsilon, like there are subdivisions which is responsible for proofreading and repair. Beta is responsible for DNA repair. Gamma is responsible for mitochondrial DNA synthesis. Because in case of eukaryotes, there are mammalian system, there is mitochondrial DNA apart from nuclear DNA also. So for this synthesis, this one helps. Then delta, like E. coli as E. coli polymerase 3, DNA polymerase 3. In case of mammalian system, it is polymerase delta, which is processive leading strand synthesis. So it helps for chain elongation. Yeah, so there are different polymerases like that. Polymerase delta is there, polymerase eta is there, and you have other polymerase like alpha. So like for initiation and elongation of DNA synthesis primer, the priming process involves nucleophilic attack by the 3-hydroxyl group of the RNA primer on the phosphate of first entering deoxynucleoside triphosphate with the splitting of a pyrophosphate. So like, and the DNA polymerase alpha is mainly responsible for synthesis of the primer. So this one is also called primase. Like DNA polymerase alpha has the property, like it is a primase also, which is responsible for synthesis of the primer, RNA primer. So the selection of proper deoxynucleotide whose terminal 3-hydroxyl group is to be attacked is dependent on the proper base pairing with the other strand of DNA. And this follows, like it always follows Watson and Crick model. Like if A is there, there will be the base pair which is coming is T, like that, ATGC. When an adenine, like it is the same thing. By the stepwise process, the template dictates which deoxyribonucleotide triphosphate is complementary, and by hydrogen bonding, it holds in its place while 3-hydroxyl group of the growing strand attacks and incorporates the new nucleotide into the. So it is always a 3' hydroxyl group of the growing strand which attacks, and that's why it always moves in 3' direction. Yeah, it's like this. Yeah, A always there. This is the complementary AT. So if it is A, T will be there incorporated. If it is T here, A is incorporated. So like the proofreading action is there when there is any mismatch here, there will be proofreading. So DNA polymerase goes for that. And then you have DNA topoisomerases, which are responsible for relief of supercoils, which is done by topoisomerases. There are 2 types of topoisomerases. One is topoisomerase 1, which acts by making a transient single cut in the backbone of the DNA, enabling the strands to swivel around each other to remove the buildup of twists. So that this topoisomerase 1 acts by making a transient single cut. So it makes only a single cut, which removes the twists. So there won't be any twists in between. Then you have topoisomerase 2, which is called DNA gyrase also, which acts by introducing double-stranded breaks. So there is only single cut by DNA topoisomerase 1, where there are 2 cuts which are made by topoisomerase, double-stranded breaks, enabling one double-stranded DNA to pass over another, thereby removing knots and entanglements that can form within and between DNA molecules. So this doesn't allow entangling of the DNA strands. Otherwise, like because it is a super helical structure, there is every possibility that it might get entangled. So it helps for the DNA molecule not to get entangled. Okay. So there is like a single stranded cut, and there is topoisomerase 1, and there is like, it is like that. The nick is like, there is a transient this thing. So there won't be any this thing. So like primer removal and nick sealing. Primers are removed by DNA polymerases by replacing ribonucleotides with deoxyribonucleotides. So these primers, they with the help of DNA polymerases, they are removed, and the ribonucleotides are replaced by deoxyribonucleotides because it is a DNA. Nicks are sealed by DNA ligases, and multiple primers on the lagging strand by a single. There is like this, you will know. And then proofreading and editing. There are 1,000 base pairs per second. So there might be every possibility that some errors are there. So DNA polymerase 1, it proofreads and corrects the typos. It repairs the mismatched bases. It removes abnormal bases, repairs and damages throughout the life. So DNA polymerase 1, in case of E. coli, it reduces the error rate from 1 to 1 in 10,000 to 1 in 1,000 million bases. So like, see, it's such a complex system, but because of the like proper functioning of the genes and all, the things will be in a proper shape. There won't be mismatches and all. Then how this termination of replication takes place. In case of prokaryotes, DNA replication terminates when replication fork reaches a specific termination sites. Because it is very simple in case of prokaryotes, only one replication bubble is formed. So whenever it reaches specific termination site, it gets stopped. The two replication forks meet each other on the opposite end of the parental circular DNA. So this process is completed within about 30 minutes, a replication rate of 3 into 10 to the power 5 base pair per minute in prokaryotes. So this is very fast. DNA replication is quite fast in case of prokaryotes, which is only 30 minutes time it takes. The entire mammalian genome replicates in approximately 9 hours. So because the genome size is so big in case of mammals, it takes 9 hours to replicate the complete genome. The average period required for formation of a tetraploid genome from a diploid genome is in a replicating cell. So it gets duplicated. Again, it will be in daughter cells, the half will be divided. So again, it comes. And how this termination takes place in eukaryotes. So you have this replisome where you have polymerase eta, polymerase alpha, and polymerase eta. And these are the helicases. This is the fork direction. Okay, how the DNA fork is moving. Now, the process goes on like this. And there is like inactive polymerase eta, then polymerase delta to polymerase eta. There is collision release and all. So there is some reconfirmation of these things, and there is termination like eta to delta hands off on leading strand. So this, in case of this leading strand, there is hands off on leading strand, and the chain termination, how it takes place here. The polymerase eta, a polymerase delta synthesized on all nascent strands. Here what happens, a switch from polymerase eta to polymerase delta on the leading strand during replication termination, thus converging replisome must undergo some reconfirmation or disassembly before encountering in order to allow polymerase delta to take over synthesis on both the strands. So there is a reconfirmation, and the function on this changes. So there are some studies, actually it is not much work, like people are still working on some of the things, are not very clear. So there is like a switch from polymerase eta to polymerase delta on the leading strands during replication termination, and there is a reconfirmation or disassembly before it encounters such process. So this leads to termination of replication. So the chromatin structure must be reformed after replication. So once replication is over, the chromatin has to be reformed. the newly replicated DNA is rapidly assembled into nucleosomes, because at the time of synthesis, when DNA replication is taking place, that time it is like all these, the nucleosome thing goes off, and there is single stranded DNA, and all the histones are removed from that. So again, like there will be reconstitution of chromatin structure. So there is assembly of, they get assembled into nucleosome, and the pre-existing and newly assembled histone octamers are randomly distributed to each arm of the replication fork, and then it goes for again supercoiling. The DNA synthesis and cell cycle. Uh, so in animal cells, including human cells, the replication of DNA genome occurs only during the synthesis phase. Okay, like G1 phase, there is preparation for that. All the like enzymes and all which are required for that, they just start getting assembled. And this is the synthesis phase where the replication takes place. This is usually temporarily separated from the mitotic phase by non-synthetic period referred to as gap phase, and get like this is G2 phase, which like goes for mitosis after that. The cell prepares for DNA synthesis in G1 and for mitosis in G2 phase. Then you have telomeres, no? Like, I think if you attended my earlier classes, you have idea about telomeres. So what happens in eukaryotic replication, following removal of DNA primer, RNA primer from the 5' end of the lagging strand, a gap is left. Okay. So this gap exposes DNA strand to attack of 5' and exonucleases. This problem is overcome by telomerase. Like always, whenever replication takes place, so when the primer is RNA primer is removed from the 5' end of the lagging strand, then a gap is left always. So this, so if exonucleases, they work on this gap, what will happen? The bases will be degraded. The DNA will be degraded. So many of the genes which are useful, they will be removed from there. So to protect the DNA from getting degraded, telomeres are there basically. So telomerase enzyme, it helps for that. So repeating non-coding sequences, as I told you earlier, that there are repeating bases in telomeres. Okay, these are the repeats which are there in the telomeres. So like this is a protective gap as such. So this in the lagging strand, there is this gets exposed here. Okay, and the telomerase is what it does. The enzyme synthesizes TTAGGG and repeats on to the telomere sequences using internal RNA template. So it uses the internal RNA template here, and this telomerase is responsible for synthesis of these repeats so that the DNA gets protected. Like otherwise these ends where there is gap and all, they will be degraded by the action of nucleases. So the telomerase, it helps a lot for protecting these gaps. This one is. So there is like the telomerase acts like a reverse transcriptase. It recognizes 3' end of the telomere, this 3' end, and based on RNA component, a small DNA strand is synthesized here. So slowly there is synthesis of these repeats, and the cap will be protected with the telomerase. So this is also the action, like how telomerases act primase, because this act from here only. The RNA primer is from the 3' end only. The RNA serves as a primer for DNA polymerase, and this DNA polymerase, again there will be synthesis. This telomerase, different level of activity in different cells. See, because as I told you, with the aging and all, the action of telomerase, like because of that, the gap size gets shortened. So we can know the age of a person, and because of that only there is more of aging and all. And there are many activities which get diminished because of the because we lose every division, we lose lots of bases. So different level of activity in different cells for telomerases. There is high activity in stem cells and cancer cells. Activity is lost in old age, and the potential target for new anti-cancer drugs. Actually, this is the potential target because what happens? The activity of telomerase is very high in stem cells and cancers. So like people can, when they are interested for synthesizing the drugs and all anti-cancer drugs, they can see that because telomerase activity is high in these cells, they can target it. So this is about how DNA replication takes place. So like there is unwinding, it forms replication fork. Primase synthesizes RNA primer, continuous synthesis in leading strand and discontinuous synthesis in lagging strand or Okazaki fragments. And synthesis always goes for 5' to 3' direction. Primers are removed, nicks are sealed, proofreading activity by DNA polymerases. And again, it gets reorganized into chromatin structure. So now the next question is, how to ensure initiation of replication only once per cycle? See, when it has to go for DNA replication, so what are the control measures which see to it that DNA divides only once per cycle? Because if it divides continuously, like again and again, twice or thrice, then the number will change. There will be like the ploidy will change. So like what are the different control measures which are responsible for ensuring that the DNA replication takes place only once in each cycle? So there are different controls. So control of DNA replication at ORIC is complex here. There are 3 levels of control. One is like this example is for prokaryotes because DNA is there in prokaryotes only. But as such, the basic process is same. There is ATP hydrolysis by beta clamp. Okay, so this is the ORC. There are 3 levels of controls. Other one is sequestration of by a protein called A that binds hemimethylated DNA in ORC. Then titration of DNA A and DNA A locus lowers the level of free DNA A. Like what happens in beta clamp, there is a beta clamp here. So there is ATP hydrolysis. This is one level of control. The other level is there is sequestration by Seq-A. And then this is like titration of DNA A. So I'll tell you these things now. So what happens, like you will be able to understand the things in a better way. So methylated DNA in the origin can be distinguished from the replicated DNA. So what happens in ORIC, there are 11 copies of GATC. So this replication of methylated DNA gives hemimethylated DNA, which maintains in state these sites until the demethylase restores the fully methylated condition. So always the parental, how you can demarcate the parental strand and the newly formed synthesized strand. So parental strands are always methylated strands, okay? So there is like these base pairs are there. There are 11 copies of GATC as I told you. So here it gets methylated here. So parental strand, this is the methylated DNA, okay? Where methyl group is attached here. So when replication takes place, it is in hemimethylated condition because the parental strand is methylated and the newly synthesized strand, it is not methylated. So you can demarcate between the parental strand where both the strands are methylated. Here, in case of the daughter strands, like one is methylated, the newly synthesized strain, it is not methylated. So like there is an enzyme called DNA adenine methylase, demethylase, which is responsible for methylation of these DNA strands, daughter strands. So daughter strands with hemimethylated DNA, we call this condition as hemimethylated condition, okay? These are, because both are methylated, we call it methylated condition. So daughter strands with hemimethylated DNA cannot be used to initiate a replication cycle. So the next cycle, why it doesn't take place? Because these this DNA, which is hemimethylated condition, they cannot go for DNA replication once again. So this is a positive control that demethylase, it doesn't go for methylation of the strands faster. It delays remethylation in ORIC so that the replication will be stopped or it will be delayed. Okay, this is one type of control. So methylation of your newly synthesized DNA with the help of demethylase it will stop replication of, so there won't be any replication unless until it gets methylated. So it is like that. Only fully methylated origin can initiate replication. Hemimethylated daughter origins cannot be used again until they have restored to the fully methylated. So this is your active origin where methylated DNAs are there. Replication, normal replication takes place. And inactive origins are like that. The parental strand is methylated. Then demethylase, like it has to methylate this one where GATC sites are there, and then it again becomes active origin. So if it is delayed replication, now it is this active origin again, it is response, it can go for DNA replication once again. So that one is a control for that. This is like, this site is where methylation takes place, again with the help of DNA methylase. This is active origin replication when it takes place, like only one strand is methylated. This is inactive origin. So like only demethylase has to function, and then other thing is sequestration. The membrane bound inhibitor binds to hemimethylated DNA. So a membrane bound inhibitor binds to hemimethylated DNA at the origin and may function by preventing the binding of DNA. This example is in case of prokaryotes, but in eukaryotes also it might be similar, where there are other proteins which are responsible for responsible for initiation of the process. So here what happens, there are some inhibitors, membrane bound inhibitors, which bind to this hemimethylated DNA, and they prevent the binding of DNA A. So that DNA A, because DNA A binding is very much necessary for replication. So what happens here, this inhibitor, it binds to hemimethylated DNA here, so that it will not go for further methylation and all. It cannot be, it cannot go for DNA replication. So this is called sequestration. DNA is remethylated and releases the inhibitor. So once remethylation takes place again, the inhibitor is removed, and then it can go for replication. Like again, these proteins bind and all. So when it has to be stopped, that time the inhibitor, it binds to the membrane. Okay, the membrane bound inhibitor binds to hemimethylated DNA. Remethylated DNA inhibitor is released. Then again, this inhibitor is released from here, and then it gets remethylated. The DNA A binds to this, and then replication will be initiated, which will take place in the next cycle, not in the same cycle. Yeah, this is like to show you methylation state of DNA may regulate replication. So there is a membrane bound inhibitor which competes with DNA A for this origin of replication. And this is hemimethylated DNA. This is activated, same thing. So demethylase, its action is delayed, and where it takes 13 minutes, and DNA promoter repressed also has delayed methylation, where in comparison 1.5 minutes, which is very fast when there is active origin. So how to control the multiple replicons in activated only? Is activated only in once time. Like these are some of the things, positive controls. There are some more things which can also control that only once replication takes place. It gets activated only once. So there are some more things which help for that. There are by rate limiting component which function only once at the origin licensing factor. So there are some licensing factors which are also responsible for seeing to it that the DNA replication takes only once per cycle. So there are two purposes of these licensing factors. One is to prevent more than one cycle of replication by removing the component. So makes the replication initiation dependent on cell division. So what happens? These licensing factors in the nucleus is inactivated after replication. So once the replication takes place, these licensing factors, they get inactivated. A new supply of licensing factor can enter only when the nuclear membrane breaks down at mitosis. So it will take place like before mitosis. During synthesis phase, only these licensing factors are there in the nucleus. Okay, so prior to replication, nucleus contains active licensing factors. So this is the these licensing factors are present in the nucleus before it goes for replication. After replication, the licensing factor in the nucleus is inactive. Licensing factor in cytoplasm cannot enter nucleus. Okay, it is present in the nucleus before replication. Once the replication takes place, it comes to cytoplasm. Okay, there is no licensing factor in the nucleus. So what happens that it doesn't allow replication to take place. And then dissolution of nuclear membrane during mitosis allows licensing factors to associate with nuclear material. So once cell division takes place, then again nuclear membrane during mitosis will allow this licensing factor so that it can, cell division generates daughter nuclei component to support replication. So once it is again divided, then the daughter cells will have licensing factors so that once the cell enters the S phase, again there will be DNA replication. So there are licensing factors which are present in the yeast. Like you have origin recognition complex, which binds to A and B1 in ARS. CDC6, a highly unstable protein whose life cycle is only, half-life is 5 minutes, synthesis only in G1 phase. So this kind of proteins, they are licensing factors which are very unstable proteins. So their synthesis only takes place in G1 phase. Okay, like as you have seen in the first, I think anyway I'll send you these things so that you can brush up yourself and you can like get the things properly. With one class, I think you can't get it properly. So you can just go through and you will understand it in a better way. So what happens, these things will be synthesized only during G1 phase because the cell will enter into S phase. That time it will be ready for replication. So this CDCX6 will allow these mini chromosome maintenance proteins to bind to the chromosome. And then when the replication initiation takes place, all these things are licensing factors. Okay, as you have seen in the beginning, when replication before going for replication, there is assembly of all these proteins. So these are the proteins which are also called licensing factors, and they, as replication initiation takes place, they will be displaced. It's like this. See, these proteins at the origin of control susceptibility to the initiation. So origin recognition complex. So when during early G1, this is the origin of replication here, origin replicating complex. These are some of the things which are already there. So this CDC6, it comes during G1 phase only, and it makes a pre-replication complex. Then it allows these mini chromosome binding maintenance proteins. This also binds here. And now this, after that, it enters here for S phase, because here it is getting ready for replication. So during S phase, when the replication, this is pre-replication complex, and in post-replication complex, these proteins are then removed from here, and then like the DNA replication takes place. And G2 stage, again, this is the same thing. So again, when the cell enters after mitosis, when it enters the G1 phase again, so again, these things will go for, it will help for replication. And once the replication is over, again, this kind of situation comes. So this is about the positive control of replication. Each origin of replication must be bound by an origin recognition complex of proteins. These remain on the DNA throughout the process. Then accessory proteins called licensing factors. See, other proteins are already there. And then these licensing factors, like as I told you, CDC6 or MCMs and all, these accumulate in the nucleus during G1 phase of the cycle. They include, these are called licensing factors, CDC6, CDT1, which bind to the ORC and are essential for coating the DNA with MCM proteins. So all of them are called licensing factors, which are present in the nucleus only during G1 phase. Okay, only DNA coated with MCM proteins can be replicated. So once replication begins in S phase, CDC and all this licensing factors, they leave like either they go for ubiquination. Ubiquination, I think you all know that the proteins will be degraded by adding a ubiquitin tail, and it gets destructed in proteosomes. So proteosomes, where the proteins get all it gets degraded there. So ubiquination is the process of degradation of these proteins. So they just get degraded. The MCM proteins leave in front of the advancing replication fork. So all these licensing factors, they are present only till S phase in the nucleus. Once the cell, then they go to cytoplasm afterwards in G2 phase. And once the mitosis takes place, again, it will be coming to the nucleus in daughter cells. So that is about the positive control of which I told you till now was positive control of replication. How the replication is controlled in such a way so that there is only one cycle. Okay, it doesn't go for repeated cycles. Otherwise, there will be all problems, like there will be all manifestation, disease conditions and all. So there is another control which is called negative control of replication. So there is a protein called geminin. This protein, this G2 nuclei also contain at least one protein called geminin. So after replication is over in G2 nuclei, because there's after S phase, you have G2 phase, that prevents assembly of MCM proteins on freshly synthesized DNA, probably by blocking the action of CDT. So this geminin, it prevents, that's why it is called a negative regulation, because it doesn't allow the assembly of MCM proteins on freshly synthesized DNA and also blocks CDT1 and all. If you recollect the first few slides, all these proteins, they get accumulated before and they may get accumulated there so that the helicases start acting there. As the cell completes mitosis, geminin is degraded so that DNA of the two daughter cells will be able to respond to licensing factors and be able to replicate their DNA at the next S phase. So this will be there in the G2 phase only. Once the mitosis takes place, so it doesn't allow, that's why it is called a negative control of DNA replication, because this geminin protein, it doesn't allow the assembly of these proteins. Okay, so unless until there is assembly of these proteins, it will not go for replication. So after mitosis is over, it gets degraded so that both the daughter cells, they will be responding to the licensing factors, which are like CD6, C6 and all other proteins. So what is like the fidelity of DNA replication? So there is, anyway, because there is proofreading activity by DNA polymerases, one mistake per billion nucleotides might take place. How this incredible accuracy is achieved? This is because of proofreading of DNA polymerases, or there is mismatch repair. This already I told you, like DNA polymerase removes nucleotides from a DNA chain once at a time because of its exonuclease activity. So exonuclease activity improves the accuracy of DNA polymerases by allowing the polymerase to back up and remove the last nucleotide inserted into the DNA chain if it is a wrong nucleotide. So proofreading takes place like if there is insertion of any of the wrong nucleotide, it removes it. The polymerization activity then puts it in the correct position, like the correct nucleotide, and continue, then elongation of the chain continues. The combination of these two activities of DNA polymerase make the copying of DNA very accurate. So it is like quite, the process is like, there won't be any error-proof process. So this is proofreading activity, incorporation of G in place of A. So if such thing happens, where like here in place of C, T requires A. So there is G here. So the polymerase exercises mismatch G via 3 to 5 exonuclease. It removes the G from here, and then there will be insertion of synthesis precedes with incorporation of A correct base. So here again, there will be A in place of G. So such kind of, if any mistake takes place, it will be removed. This will be exercised with the action of exonuclease. And then again, there will be incorporation. So these polymerases, they help for proofreading activity. So this is about DNA replication. I hope you people could understand the process. Already, I think you people have studied it, but there might be some things which you can. This is like, because all your molecular biology is the thing which you have already studied somewhere, but the things are same. There are some additions to that. Okay. So with this, I'm completing the class now. So next class, I'll tell you like whenever I'm going for the class. Fine. And I